Okay, so I'm Sam. I think a lot of people know me already, but I'm a PhD student in MCSB in the Swarup lab. So I mostly do like single cell and spatial transcriptomics and epigenomics in Alzheimer's disease and other brain things. But today I've been requested specifically to give a talk on data visualization. I think because people like aesthetically like my figures that I make, but that doesn't necessarily make me an expert on any of this. So this, and like I think data visualization in particular is like subjective, and there's not one right way to do it. So this is like, I will just warn you that it's a very opinionated talk. And this is like someone, so I guess that kind of leads me to the first thing is like, I believe that we are all sinners and we've all made bad graphs, and there's always better that we can do, right? So I'm not going to give you like the boilerplate guide to making perfect graphs every time.
Do you want back and forth with
Absolutely.
this talk?
Yeah.
Or do you want to just talk?
No, you can do back and forth. So I think, if you're like doing data analysis, you probably like generated like tens or hundreds of panels, but then finally when it's time to write your paper, like how do you go from like individual panels to I mean, whether or not you think this is a nice figure is subjective also, but to like a nice published figure, right? That has like many different panels and like lots of different colors and labels. How do you actually accomplish something like this? It's typically not something that's taught in like grad school courses or anything. A lot of people kind of like have to figure this out as they go. What are you looking at, Emmanuel?
Like, what is WNN you have?
Weighted nearest neighbor. Don't derail me for that. Okay. I first want to just establish like the importance of data visualization. So there's this paper from a couple years ago in genome biology titled "A Hypothesis of Liability," and it's I guess it's an editorial opinion paper on like the strengths of doing like genomics analysis where we don't always have a direct hypothesis going in. So they have this really nice example, I think, where they gave these like data science students this one data set that basically it's not a genomics data set. It's very simple. It contains body mass index and the number of steps taken per day, as well as the steps for like 1,000 subjects or something. And then the students were split into two groups where one group was kind of given no hypothesis. They're like, here's a data set and find some patterns. Okay. The other groups are given these three hypotheses, which is there a statistically significant difference in the average number of steps taken by men and women? Is there a negative correlation between BMI and number of steps for women? Is there a positive correlation in men? So to answer these three questions, you don't really need to do any data visualization, right? But what happens when you plot the BMI by the number of steps taken is you get this graph, which is just a gorilla waving at you, right? So there actually might be some statistically, I mean, this is like a fake data, just for full clarity, this is not real data. But the point being that when you have a hypothesis, the hypothesis focus group, 14 out of 19 did not find the gorilla because they didn't plot the data, right? And some of the hypothesis-free groups still didn't plot the data. I think possibly because it's just like a pretty small data set. They might have just kind of run the correlations and test that the hypothesis group did anyways, but more of them did end up finding the gorilla by plotting the data. Yeah, so I like this example a lot, and it's a good motivation for like why you should do data visualization. But I think a takeaway from this is that good or bad or no data viz can greatly alter our perception of the data, and it could be used in a misleading way if you're a bad data visualizer. So the next part of the talk, I'm just going to talk about tools that we can use for data visualization, and I'll only really focus on R and Python tools. And I'm just going to give like a broad overview, but to skip to the conclusion, I guess my suggestion is just to basically master one of these and have a working knowledge of another one or more than one. It's especially useful if you are doing genomic stuff and like a little bit of R and a little bit of Python. So like, yeah, so the main Python library is called matplotlib. And the name is actually because it was originally trying to copy like the plotting functions that are built into MATLAB. So that's why it's matplot. But this is just from their documentation site, their own description. It's a comprehensive graph library for making static, animated, and interactive visualizations in Python. Their website has tons and tons of demos and tutorials for making different plots. So I'll just like blast through their gallery where like if you're like, oh, I want to make a bar plot, you can click on this link and it'll show you the code to make the bar plot. And there's all sorts of different plots. So you can make like dot plots, line plots. You can make these box and whisker plots, violin plots, histograms, violin plots, histograms, blah, 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 blah. A lot of these have like kind of different flavors where you'd want like to group them in different ways. So these tutorials, and there's more that I'm not even showing. So there's like plenty of tutorials to get you where you need to go on the matplotlib documentation site. They also have a lot of different tutorials for just picking color schemes and styling your plots. And then there is a very popular add-on, or I guess extension of matplotlib that is, it is like technically a, like it's like a super class of matplotlib, if that makes sense. So it's built on matplotlib, but it's their own plotting functions, and some of them are like redundant in a weird way. But this is like a snapshot of the Seaborn graph library or the gallery, where for each of these like panels, you can click on it and it'll show you the code for how to make it with an example data set. And very similarly, there's something called the R graph gallery, which is, yes.
Can I just ask an important question?
Yep.
For what's the primary like difference between Seaborn and Matplotlib, and why would you stick using Seaborn over?
I actually have no idea.
Okay, so I'll that was a leading question. So I personally use Seaborn more frequently because it is naturally compatible with Pandas data frames. So that is like the one primary reason why I use Seaborn. And to do a lot of tuning, I do have to use Matplotlib. But you can group and hack your data frame by different variables that are already in it, provided it's a clean data frame. So that's why I use it.
Cool.
Yeah.
Thank you for letting me derail you.
Thank you for that
I've
comment.
also found that the nice thing about Seaborn is it kind of forces you to think about like what you're doing in a way that Matplotlib tends to not. So like, for example, it forces you to think like, are these categories or are these continuous variables? Like, should this be a cat plot? Should this be a rel plot? Stuff like this. So I really like Seaborn. I've found bugs within Seaborn, but it's a really nice package. I will also say that the plots are typically more aesthetically pleasing, like base plots compared to Matplotlib, at least in my experience.
Cool. So, okay. The next slide is just kind of showing that there's a similar graph gallery in R, called the R graph gallery. But this one typically includes examples from both base R and ggplot2. And I think like out of all these tools, ggplot2 is a very like fundamentally different type of plotting where it's something called like the GG stands for grammar of graphics, where you start with your data set and then you like add one piece at a time. So if you're like, I want a scatter plot, then the dots will show up. And then if you're like, okay, now I want a regression line, the regression line will show up. But if you do it the other way around, the regression line will show up first and underneath the dots. So it's very, very like clear as to like what gets added onto the plot at what point. And for that reason, I really, really like ggplot. And it has like so many add-ons now that other people have developed that I think like you can make like literally any plot with ggplot. And there is so Matplotlib and Seaborn don't use this grammar of graphics type of thing. They're usually like, here's a scatter plot function. And then it could be really difficult to like get a customized plot that has different layers on it.
You can do the same.
I think it's way less easy to do it, and it's not as like principled as the grammar of graphics. But there is a newer grammar of graphics library in Python called plot9 that I have not used because it has I've tried to install it in my ScanPy conda environment, but it has conflicting dependencies. So I'm like, all right, I guess that's not happening today. So yeah, like my recommendation is basically to pick one of these, and like you should master it to the point where you can make, I would say you can make like simple plots without looking anything up. I mean, that could take like years, honestly. But and then like if you're like, oh, I want to add this, then you should know like what documentation. So like being familiar with documentation, making a lot of different plots, you'll eventually master one of these. Like for example, I would say like I'm like mastered ggplot and I'm like pretty proficient with Matplotlib and Seaborn. I would love to try plot9. Plotly is another interesting one, but it is more like it's like mostly for interactive plots where you can like hover over points and it'll like show you different things, or you can like zoom in. And Plotly has like this web integration where if you have like a paid plan, your plots will just like show up online on your like profile, and you can like easily share them. So I think it's kind of, I don't see a lot of like academics using Plotly, but I think a lot of people in like data science industry do it.
It's also got very, very poor documentation.
Yeah.
Experience.
Plotly?
Yes.
Plotly is interesting though, because it's multi-language. So there's an R, Python, there's a JavaScript, there's a lot of different ones. And they do have like, if anyone's heard of like Shiny, which is an R, it's like an R data science website type of thing where you can like have interactive plots. Plotly has something called Dash, which is basically the Python version of that. Yeah. So for like, I would recommend these two books for learning. Like each of these books have chapters on data visualization, but they're just generally data science books, and they're both free. The R for data science one is really good. It's from Hadley Wickham, who is like the guy who made the tidyverse, which is like kind of a mini language inside of R, that is really good for handling data. And I don't know as much about this Python data science book, but I did like look through it before like putting it here as his recommendation. And it's like each of the chapters has basically a Jupyter notebook that has code and text blocks, so it's explaining what it's doing. And I think you could, if you like know a little bit, you could probably skip straight to the data visualization chapter, and it should like give you a basic primer on Matplotlib and hopefully Seaborn. Okay. So the next thing, now that we know some of the tools, is how do we actually go about constructing complex figures for publications that are made of multiple panels? So in Matplotlib, you can do this thing called subplots, where you can kind of like set up this grid, for example, like this 2x2 grid. If you have 4 plots, you can be like, okay, plot 1 goes in coordinate 0, 0, plot 2 goes here, 3 and 4, and so on. And you can get pretty crazy with the layouts too. If you like, for example, this A, C, B, D thing, with different size plots in different parts of this grid. And this is nice. And R has something similar called patchwork, where you can set up these like kind of coordinates and then define where each plot goes and how big they are and all of that. I believe patchwork works with ggplots as well as the base R plots, which is nice too. So I think that for more complex tasks, it's like you always end up having to go for like a post-processing, once you've like generated like all your figures, like a post-processing program. Typically, I don't know if there's others, but I'm aware of Adobe Illustrator and Inkscape. Adobe Illustrator is like the premium option because it's like it's not free, but I believe UCI, I don't know if it's still true. Does UCI still pay for it for us? They don't? Okay, so I can't really recommend it.
My recommendation is something called Affinity.
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 Inkscape is also free.
Yeah, Inkspace is similar to Illustrator and it's great. It has all the functionality that you have in Illustrator.
Cool. Thanks, Nargis.
Yeah, I mean, I like Affinity a lot. It has everything that I found that you need for this. It's not free, you need to pay for it.
Okay.
But unlike Illustrator, you pay for it once.
Okay. Yeah. So Illustrator is a subscription, and like it used to be like, I don't know, UCI like randomly, it seems like, decides to pay for it and then they take it away. Okay, so I mean, it seems like, do people's PIs pay for it? Is that common? Yeah, yeah. In the neurobiology department. So it's very case by case, obviously.
It's like 100. Yeah, if you pay for it.
If you're a student.
All creative, now your PI is like you go through.
Okay. So I would like, I think when I started as a PhD student, I like told my boss, I'm like, I don't know how to do anything but Illustrator. So if you want me to make figures, you gotta like get me a license. I probably didn't say it like that, but you know what I mean. So I mean, hopefully they'll shell out the money for you to get a license. And I don't think you should pay. If they won't, I don't think as a student or a postdoc you should pay for it for yourself. So I would just try to do something like Inkscape. But basically, like, this is just an example of like the workspace with a figure that I recently made, where like obviously there's like a ton of different charts on here. Each one of these is a ggplot object that I like saved as its own PDF, and I had to like go in and assemble them. And this is kind of like a lot of trial and error of like how it's almost like, I would think of it like Tetris, where you're like trying to fit all the pieces in a way that it makes sense. So there's not like these big like gaping holes or like things aren't lined up right. Like in this figure, I think I'll go on the next slide, but I'm getting ahead of myself. So I think that what you can do with Illustrator is kind of like, if you save your plots as a PDF, you can easily change like different attributes of the plot. So like if you want to change the color, you can like select all of this color and then change to that color. If you want to change the line thickness so they're all the same across all your plots, very easy to do. If you want to change the fonts to be consistent and font size, also very easy to do. If you need to like add like an arrow to point at something or like add some text, you can easily do that. You can also do like digital, like I guess digital art. Like if you have an iPad, there's an Adobe app so you can like draw a cartoon if you want to have a little cartoon, like a graphical abstract or something. I also frequently will use like, I didn't put a slide for it, but there's something called like BioRender, which is basically like a library of biology images. Like it'll have a picture of a brain or DNA or a spleen or whatever, and you can like take those graphics and easily add them into Adobe to make like your little cartoons and stuff like that. Yeah.
You mentioned that you output all of your panels to PDFs.
Yes.
In my experience, that makes the actual file you're putting all these panels into like massive large.
That's like not exactly true. If you, I should have had a slide on this, but there's like at least for ggplot, there's something called ggraster where you can select which layer. Like rasterizing is basically compressing your like your image into like, or I guess compressing the vectors into one image. When I'm saying vector, like each like dot in a scatter plot, there could be like hundreds, but they're all individual objects. So if you rasterize them, they all become like one object, and it's way smaller. So for ggplot, you can say like, okay, I want to make a scatter plot and I want this to be rasterized. But when I'm adding labels, don't rasterize those. Like for these violin or volcano plots, like this label, I can easily change that, but I can't change the dots underneath. They're all one object, right? So then that makes it nice and small. But if you do try to load like a volcano plot with thousands of genes, each of these is going to render as a dot in Illustrator, and then Illustrator will be very, very unhappy. It'll make your computer slow and loud.
Another solution is you can manipulate your plots in Illustrator, load it in once. When you get it to where you want it to be, then you can rasterize it in Illustrator, and then you can embed it in your file, and it shouldn't give you as many issues.
Cool. So yeah, to go back to this plot, basically like I was trying to make like each row was kind of like grouped in a certain way for this like part on the right, and then each column also was grouped in a certain way. So like with Illustrator, it was really easy to literally use the alignment tool to be like, this object align here, and then they're perfect. And if you can also just like say the XY coordinates to make them like perfectly lined up as well. And like I guess maybe I'm just really nitpicky, but like sometimes like also this is like, to be fair, this is not my favorite, but I do think this is a good paper. But there's like some small data viz gaps in this paper that like it's like I think like in some way it's a reflection of you and the quality of work when like it's something that's permanently there, right? Like you're not going to be able to just like, oh, I'm going to change the color and then ask Nature Neuroscience if they'll update it. But this one is like in this heat map, or sorry, I guess like this is kind of similar to my plot where I was like trying to align things, but they're like doing this vertical alignment, and they have like these different cell types. There's 9 of them here. And then at the top, they're showing this like proportion analysis in their different conditions. And then the middle, they're showing this like gene expression heat map. The bottom, they're showing this gene expression violin plot. But for some, I don't know how this happened, but like on the very leftmost, like in the heat map, the very leftmost column is like a little bit wider than the rest of them. So then when you go down to this one, it's not like the one that I boxed is, you can see this is not lined up, right? So like this is like in the middle of 2, and it's like you're doing this like mental math to line them up afterwards. And I think that the mistakes like this can be made pretty easily in Adobe actually when you accidentally drag something over. But this happened like to 2 of their figures that are different. So I'm like, I don't know how this possibly happened, but be really careful when you're like lining things up, and this could be easily avoided if you're using something like Adobe. Another thing I'll recommend is like, I guess we already talked about this a little bit, but use vectorized plots or high quality images. This is a paper that was published like 2 weeks ago in Cell, and you can see this like kind of visual artifact crap all over their UMAP plot and in their box plots too. But like, so I don't know, this could have happened at many different stages, right? Because you usually are supposed to give, like, when your paper's getting published, you're supposed to give them the like PDFs or the Adobe files, and then they're supposed to like typeset it. But then, you know, right before it's published, they give you a proof, which is like, it's like the paper typeset, but it's not published yet, and you're supposed to check over it. But like in the proof, they could have seen, oh, this is a low quality image. They can ask the editor to swap it out, right? So you got to be really careful. I mean, first of all, you should provide the high quality PDFs, but the mistake could have been made on the side of the journal also, right? But now this is not going to get updated. This is how it is now. And like, this is just what the paper is forever. And it could be a nice paper, but like, I don't know, maybe I'm just super picky, but I think I'm like trying to understand a paper. I'm like, wow, why is this figure like so low quality? It's just distracting, I think. And then I guess like as a side note for this, if you're like taking screenshots or figures for a PowerPoint, I've seen a lot of time when people have like a really low quality image in there. And I think that's like not as bad as it being in a paper, but I think it's like you should go and find, like, typically if you go to the web browser version of a paper, you can click on the figure and then it's like the full high quality resolution, and you can just copy that and paste it in your PowerPoint, and then there's no garbage. Like, but like even for this one, this is what it looks like as the high quality one. Anyways, another thing in terms of laying out, oh, sorry, go ahead.
Can I add one thing? Sorry. Yeah, so one thing you should be careful about it. Even though when you use the vectorized plot or rasterized plot, but the size is really big, sometimes Illustrator do some funky things that just lower down your like resolutions of your pictures. So you also need to be careful. Sometimes you need to like group it, like you like save it as a PNG and copy and paste it like high quality PNG or JPEG. When you have like too many dots or have too many lines, like.
Yeah, I agree with Nargis. I think like something I do, like, this is just like a personal thing, but when I make figures, I have another device that I'll check, a totally different device than the one I'm like making the figure on, to check that the figure looks good. So like I have an iPad, so I will just like look at my iPad and be like, okay, now it looks good. That's maybe a little extra, but it is like double checking that the figure you know, isn't like low quality for some reason. Because yeah, like Nargis was saying, Adobe Illustrator can sometimes introduce weird artifacts.
What you do is you download the whole figure as a PDF and check on that.
Yeah, like I wouldn't do that after every change I make, but like if I'm like, this is the final version, then I'll like do that, and I'm like, oh wait, no, this is messed up, I gotta go fix it. Okay, also the next thing is maybe fully my opinion, but like I think we should try to like be smart about like white space, like avoiding unnecessary white space. And that could mean multiple things, right? Like if you have like I have this box here, which is just like an empty, this figure is kind of set up like a grid, but there's just like this empty zone in the bottom left. Whether or not that's, I don't know, it could be fine, but maybe they could have just added like another plot there, or they could have resized some of their plots to make it take up the full space. Because for some reason, maybe it's just me, but my eye is drawn to the blank white space. And like if you're having like, the other thing with white space is like one of the things you can do to like avoid more white space is just make text sizes bigger, which is also helpful so people can read stuff. I guess like text sizes don't matter as much for a journal figure, because people typically read them on digital devices so they can zoom in. But for a PowerPoint presentation, I think it's important to have a big enough font size that people can read them.
Journals don't like it when you give white space. Like figures.
Yeah.
They will.
Well, I don't know. Like I guess like they don't always, and it's not. I think that if you have to have it, I think putting it in the corner isn't the worst place. But maybe a slight nitpick would be put it in the bottom right corner, because that's, if you're reading like top down, left, right. I think is more natural to have it at the bottom right corner as the blank white space. And this one, like I was kind of like before I was saying like, oh, we should kind of set up like a grid, and this one is like almost a grid, where like this is like a little bit like taller than this part over here, and that slightly bothers me. But
I mean, all they could have done there is just shrunk the UMAP and then made panel C a little bit bigger.
Yeah, so like this isn't, I guess this is where like data viz becomes an iterative process, because typically I'll make like a ton of panels and then like interpret the data, and then when it's like, all right, time to write a paper, I almost always have to remake like a lot of those plots because I'm like, oh, this is a little too small or a little too big. And sometimes it's just easier to remake it in R or Python than to like mess with everything in Adobe. I'll usually try to get it like 90% of the way there in Adobe, in a Python or R, and then move it over.
Oh, and the other recommendation I would have actually is try to make your plots as close to publication ready as you can while you are doing your data analysis. Like just make that a habit. That's
I agree.
So that really helps you down.
Yeah.
You should emphasize that again.
Yeah, I can say that. Manuel is just recommending for when you're making plots with Python or R or whatever to make them as close to publication ready as possible. I think a lot of the time that means don't make your font sizes too small.
Yeah, basically. Dude.
A lot of times, I guess like even this is like pretty crowded down here, but you can imagine this heat map right here. It probably used to have like every gene labeled, and they picked the ones here that they showed over here at the like in the chromatin accessibility to match. But that, it would have been really crowded and hard to interpret if there was every single gene really small. Okay. This is totally a gaffe from like my first paper, which is like a problem with selecting accessible color palettes where like this was my first paper, which was mostly like, it was basically building off of my rotation project. So at that point I was like a very new grad student, and I was using a lot of code that my PI had wrote in his postdoc. And at that point I was like, here's results. And I was like rerunning code that he wrote. And I wasn't really thinking about like things like color schemes, but this one on the left is what was basically published. And it's like a red to green diverging color palette, which like, I feel like a lot of people have probably heard of red-green color blindness. So there's this like app, which I'll talk about more on, or I guess I'll just go to it now. There's this app on the, at least for Mac called, I don't understand the name. It's super weird. Sim Daltonism, which is
simulating dolphins.
Is Daltonism colorblindness? Yeah. Okay. That makes sense. But it's basically a little window you can drag around on your laptop that'll simulate what it would look like to a colorblind person. So like on the right here, this is what it looks like for this type of colorblind, which is red-green. And you can see that it's like pretty much brown on both sides. Or like they have slightly different browns, but it becomes really hard to interpret like the meaning of this the color of each edge in this network. So and this actually, so like it didn't happen to me in this paper, the editor didn't care. But in the next paper, I had like a single cell data set that I colored one of my clusters red and one green. And then after it was accepted, the editor was like, you need to change the colors to be colorblind friendly, which meant I had to remake, like after it was accepted, I had to remake all my figures, which was like painful. So just, yeah, I guess in line with what Emmanuel was saying, just from the get-go, try to be thoughtful about what color scheme you're using. I think on the things like I was showing earlier with the matplotlib like color schemes, sometimes they'll indicate which ones are colorblind friendly, but you can just always have this sim daltonism. There must be like a Windows equivalent, but I'm not sure. But you can always just double check with this kind of thing.
The other thing they want to use the grayscale so that you actually can see like the contrast of
right.
the colors as well. And because sometimes like the color can be different, but with how light the hue is, it's harder to do better color. So you can actually grayscale everything to contrast.
Right. That's a good point too. I think that sometimes like, I guess if someone was like fully like had no color receptors, there's only, I don't know. I think that it's going to be too difficult to accommodate, but I'm not sure. Anyways.
Can you repeat the grayscale comment for audio?
Yeah. What was your name? Sorry.
Nolan.
Nolan. Okay, so Nolan was commenting that in Adobe you can just convert your like you can basically do a grayscale view, so then you can see like if the the darkness is actually conveying something, but the where the like in addition to the color, which could be useful for this kind of accessibility thing as well. So I guess on the same note of color, there are a lot of different like color palettes that are in R and Python built into matplotlib and ggplot, as well as like others. I really like this one called Met Brewer, which is like somebody took a bunch of paintings from the Metropolitan Museum of Art and made a bunch of color palettes based on them. And I just like literally go through this website now and I'm like, I want this one. So they're really nice, and I feel like
Is it Python or?
it's both, yeah. And I believe the same author made one for another museum.
MoMA.
National Park.
Yeah. Oh, there's a national park one too, yeah. And but you can also like like there's something on Mac where you can just kind of like, it's called digital color meter, where you can just like hover over anything on your computer and it'll find you the hex code. So if you're like, I like this picture of a grape, you can just get the grape color and then write it down and use it for your plots. So it's pretty like easy to make. Like, I don't know, I think that there's enough resources that we should all be making like thoughtful choices with colors in data viz. Okay, I also talked about this a little bit already, but I think that we should consider the font size for whether your talk is for, or whether you're showing something for a talk or for a paper. This is from like one of the Surratt papers where I think that it was okay for a paper, but if this was like being shown in like a conference in a really big setting, like the figures or the text would be so small that it's like illegible, unless you had like opera glasses or something. So I think that they would like basically have to like double or triple the font sizes to make it legible. And then they also like didn't have, like, this is maybe another gaffe, is they didn't, this one in the middle was a different size in the top and the bottom, which just like irks me. So I don't know, something that could easily be avoided with Adobe. Another thing I have to add is don't ever like squish plots to make them fit. This is, I guess, once again, my opinion, but like, this is like, I legitimately will be the only thing I remember if I see a squish. Like if I see a circle that's like actually flattened, I'm like, it's just I think it's just distracting, right? It's distracting and it's not doing your science justice. Another thing to watch out for is that journals will have like guidelines for what like is an acceptable format for a figure. A lot of the times with the first submission, they don't care. They're like anything. But it is also sometimes a pain if you're like, oh man, I made it a little bit too wide. Now I have to move everything around. So if you have like, if you're starting to assemble your figures into Adobe or Inkscape, you should check which journals you're considering submitting it to. And if you're like in between two, maybe try to make it so it's going to work for either one. Like, for example, I think that the Cell one is like slightly wider than the Nature one. So if you like made it for Cell and then you actually are going to submit it to Nature, then you have to squish everything. You have to like, I don't know, make everything a little smaller. They'll also usually have minimum font sizes. And the one that kills me is always the maximum size of each file is like a certain size. So like if you have too many plots or like, then you have to go and vectorize or rasterize, sorry. Okay. After all this talk about doing things with Illustrator, there is like this total other way of doing it, which it sounds good, but I don't know how good it is. But there's a package in R called Plot Gardener, which was published last year in Bioinformatics. And they're kind of trying to say like, okay, it's like patchwork or subplots like on steroids, I think, where you can very like intently like lay out this like coordinate system in like actual physical units, and then you can say like this plot is this exact size and it goes right here. I think it's especially useful for something like this. So like this is their cartoon and then they're like, oh, we made this out of it. But this is showing like genomic coordinates. So like if you have it's kind of like the genome browser, right, where like you're showing different layers, but you can perfectly line it up in this. But like if this were me, I might just like make this plot in Plot Gardener, and then like I probably have another plot or two to add to this, and I might just like put that in Adobe, because I think it could easily get out of control with how complicated it is. Like this is from the homepage of their tutorial website, and this is alone is 144 lines of code, and I'm sure that they probably like tried to pare it down. I think that is really like a cool idea, and then like if you, I guess like on one hand, you can be like, oh, if I have to like change something for my paper revision, I can easily just like change it and then the final figure will be ready. But if you have a paper revision, you might add new panels or something, right? So it could totally, it might actually not be as reusable or useful as you want it to be. Another like in terms of like complexity, it's like the circular layouts kind of freak me out a little bit. The left one is from a paper that was actually published, I think today. I just saw it and I was like, wow, that's a lot going on. how many, I don't even know how many layers, like 1, 2, 3, 4, 5, 6 layers and labels. The right one is one that I made, and yeah, like I said, I don't really like these layouts, but sometimes rules are made to be broken, because basically my PI really does like the circle layout. So I was like, here you go, here's your circle plot. But I had this on a poster at a conference that I was at in the summer, and it was like a struggle to like actually convey the ideas here. I can't even imagine trying to like draw someone's attention to one of these like small dots in the middle, and they're like trying to like line it up, because a lot of the times they're basically like a rectangle heat map has been like curled up, right? So it might just be easier to show this as a square, because we're like easier to looking at coordinates in that way. Okay.
Can I also comment just on the last slide, actually not this slide, but yeah, in my experience, you can get like a panel really, really good programmatically, but as soon as you try to arrange multiple panels,
Yeah.
one thing, I have never had success with that. Yeah, I'm just not just
So for me, I can usually have some success if I'm like, I'll go back a few slides, but like this, these
Yeah.
volcano plots, I just made a grid of those. And then, but the other types of plots I wasn't gonna, because then like, you might be running so much code just to like load all of these plots into memory, and at the very end, you're putting them together. And like, for example, like one day I might do my volcano plots, and the next day I do these box plots, right? So it could be just like a tall order to run all of these at once, depending on the type of analysis you're doing. Okay, where were we? Okay, so this is like, now I want to just talk about like basically like common pitfalls, and this is mostly taken from a GitHub with this title: "Friends don't let friends make bad graphs." So this guy, who's a postdoc, I think he does like plant bioinformatics, which is pretty cool. He has this repository, which is like, this is his opinion, and I took a few of his examples that I think were good, but if you're interested, you can go check out this GitHub as well. The first one is basically, don't make violin plots when you have a really small sample size. So the left is like, oh, look how different they are. Well, first of all, they look like vases to me, or vases or whatever. And then like, if you look at the box plot, it's like, it looks pretty different, right? Like you can clearly see that there's some differences in the quartiles and the median and the means. But when you like actually look at the data points, there's like almost nothing going on, right?
So I think this is a really good example. What you will hear from most editors and reviewers nowadays is they want you to show the raw data. So
Right.
it's going to be rare that they let you get away with box plots without actually pointing to points.
Right.
Spot plots, they're just summary statistics, right? So
Yeah.
I think this is a valid point. You really do want to combine like the box plot and the scatter plot into one.
Yeah, sometimes I'll like, if it's a single cell data set, there might be too many points, and I feel like
Right.
it's just like literally a wall of points and you can't see
Yes.
anything.
Yeah.
So if anything, I would do a violin plot. Like, this is easy with ggplot, violin plot, and then the next thing, box plot. So they're like kind of stacked. And you can actually have like, in ggplot at least, there's something you can do where like you put two plots side by side, so you can have the points right like side by side with it, and then it's like not really, if you have too many things on top of each other, I guess that's a problem as well.
That's fair.
But yeah, I think like you get pretty different interpretations depending on which view of it you look.
Sorry, Sam. I think this is a really good example. This actually goes back to the gorilla example you showed us earlier, because what you're doing fundamentally in the middle there is you're calculating summary statistics
Right.
without actually, it's not really data visualization.
Yeah,
it's,
you're right.
You're visualizing the summary statistics.
That's a good point.
Right. And so I actually think this is really instructive, because first what you have to do is you plot things not using any sort of summary statistics or assumption about data. And then once you looked at it, then you can go, okay, I can calculate median. By
Exactly. Good point.
So I think that this is actually a really, really nice example.
Okay. Let me like blast through these, because I have a whole part at the end that's just about dimensionality reduction, and I want to get to that. So the second one is, don't use bidirectional palettes for unidirectional data. So this is like a graph showing gene expression. And I guess maybe the rows are different groups or something. So it's like, okay, this makes sense because it's like 0 to 100. So it's just like a unidirectional thing. This one is log fold change, so it's bidirectional, and they're using a red, white, blue palette. That makes sense. This one might be slightly confusing actually, because this one is negative and positive, but z-score is basically scaling something that starts at 0 and goes to like 100 or whatever. Like it's a scaled version of this. So having a unidirectional palette for z-score does make sense, because 0 doesn't mean 0, it just means like that's the mean actually. And then this one on the right that says down here, it's a data visualization sin, because it's like a unidirectional, like, you know, it starts at 0, goes to 100, but they're using this red, white, blue palette. This is definitely don't do this. I could see the more common mistake being like this one, which I think that I used to do this, but it definitely doesn't make sense. Okay, the other one that they're saying is basically when you have a heat map, you should think about how you're ordering rows and columns, where like the top one is like just jumbled mess, and you can actually, so I think it's showing gene expression by cells. And then the bottom one, okay, they've actually ordered the columns by cell type. And you can see that there's like expression showing up in cell type too. And there's these different patterns. The other one with heat maps is basically like, don't let outliers dominate the color scale. So like when you're picking your color scale, you might want to like cut off by 99 percentile or something, right? So you don't want like this to take up all of your color space. Where like this is like a much more informative heat map.
Biologists hate it when you do shit like that. So.
They don't like this.
No, in my experience, they
this was a comment in my paper that I had to, I don't know what to say.
No, I think it's 100% fair. It's just in my experience, people think.
Yeah,
I think it's 100%.
Okay. The other thing, this guy, I guess maybe this one's more debatable, but this guy doesn't like pie charts, and he says you should do stacked bars instead. But I think that pie charts, basically they have a limitation where they're maxing out at a certain, if you have like a lot of groups, they're bad, but if you have, like, this is only 4, so I think this isn't that good of an example. But even then, I think like donut charts, I think I like better, because you usually can like write something in the middle or like label them easier. But yeah, if you're trying to make these kind of multi-way comparisons, then I could see that stacked bars are a lot more useful. Okay, buckle your seatbelts for the next plot. This one is like, I guess this is where like the friends don't let friends from this GitHub ends, but I have like some similar ones that I kind of pulled out, where like the ways of showing overlapping sets. This is from a paper in Nature in 2012 where they sequenced the banana genome, and they were like showing some overlap with other species, I guess. And like, I can't even begin to interpret this plot, right? So like there's this rectangle species, like these 2 rectangles, there's the circle, and then there's these weird like ellipsoid things in the middle. They're like splatters or something. I don't even know. Like, how would you begin to interpret this?
So
like
it's a Venn diagram.
This is a Venn diagram. It's a multi-wave Venn diagram. The Euler plot is different because it's the size of the shape is scaled to the size of the set. I just think honestly, like maybe this is my hot take, but you shouldn't do Venn diagrams for more than 3. 3 is that's where it maxes out. They should have done this, which is an upset plot, and this maybe at first looks more complicated, but I assure you it is a lot simpler. Where like you have this grid down here, where the grid kind of denotes which sets are overlapping, right? On the left side is just the different sets that are there, and then the size of that set. The top one is showing the size of the overlap. So this one, right, the first one here is showing this is the unique set. The set of things that are unique to this and not found in other groups, and so on. And then this one down here is like, okay, these 2 overlapping, but you can, they're pretty customizable. So you could be like, I only want to show like the 3-way overlaps, or you can show a lot of specific things with upset plots. This is from a paper in bioinformatics where they were talking about their package called upset R, but I think fairly makes them in Python, right? Yeah, I've used upset R a bunch of times, and it's like, I feel like every time I try to make one, I have already forgotten how it worked in the past, and it's like a little bit confusing, but then I eventually figure it out. So upset plots are really good if you're trying to do a lot of different overlaps. Definitely better than doing something like this. I often also see these like kind of like lattices of like circles that are going in different directions, or like they might be slightly different shaped, and I just think those are just really confusing. So these are a lot more interpretable. And then yeah, I guess like if you're insistent, like this is from a recent figure that I made. But like if you're only showing like small overlaps, like 2 or 3, Venn diagrams can still be useful, but I would suggest to scale the circles to the size of the set so you can easily like, oh, I see the green one's bigger than the yellow one. And then if you also are doing like a statistical test for overlap, you can add the stats, like just annotate them. So like, yeah, this is a pretty good example of like, I made the, like these diagrams were actually totally uncolored in R. I like added the colors, I added the annotations, I added the numbers all in Adobe, and I like lined them up too. Okay, now that's like the end of the friends don't let friends. And the last part is misleading dimensionality reduction. So I think we've probably a lot of us have seen this plot, which is like from the Seurat tutorial. It's a UMAP plot where it's basically like a 2D representation of a high dimensional single cell RNA-seq data set, where each dot is a cell and they are localized in this space based on their similarity in a cell neighborhood graph that comes from PCA, which comes from scaled gene expression after you've done feature selection, and so on and so forth. So it's kind of like a lot of steps to get all the way down here. And I think people typically use it kind of as like a way to see their clusters. There's some uses of it, but I think it can be easily like, I think it's a good like bird's eye view of your data set, but it's hard to actually have like this be a conclusion. And there's a lot of ways to trick people with UMAPs and similar dimensionality reductions. One of them that I really didn't like, and yeah, these will end up in like high impact journals too. So like this is an example from Nature Genetics, which is like they like saw I think they saw like some gene expression patterns that are like known to be spatially oriented, like in their UMAP. They saw these patterns and then they just decided to add this axis here, which is like, I think that is like really, really bad. Because it's super misleading to like, if you think about the review, like the readers of Nature Genetics, a lot of them might be like, they might not be like in the weeds of this data set. So they're like, they're probably just going to believe that, oh, okay, UMAP means it's going to split things spatially, and that totally makes sense. Even if they like saw that pattern in their data, I don't think it's fair to write that on their UMAP. I'm actually going to go back because I forgot to say this, but I think like so in the default in Seurat is this like exact like the default plot is this exact theme where it has actually numbers on the axes, which I think immediately when you see numbers, you're like, oh, okay, so like this one is like twice as many UMAPs as this one. And there's actually like the numbers are completely meaningless and arbitrary. So I always just like remove the bars when I plot them and there's no numbers. That's how they did it here. So points to them for doing that. And I think that's how ScanPy works by default. It's just like a box that says UMAP. Okay. Also, like I think that sometimes like people come up with these like fancy like different dimensionality reductions that are like totally unnecessary in my opinion. So this is just like they have a new algorithm for projecting a single cell data set onto like on a different geometries. Like they can do a sphere. This is like the world map, whatever. They can do whatever shape. Which is like, okay, that's a Nature Methods paper, but this is them using it in a different paper that was published earlier this year. I think it's just like, I can't even see what's going on. I guess like they're colored by cell type or subtype, but like this isn't, UMAPs are already like questionable whether how useful they are. This is like fully like, they just thought it was like, okay, so this is like the Russia cluster or something, like, I don't know what's going on. Astrocytes are Antarctica. Yeah. Another pitfall, or what I would consider a pitfall that I commonly see in like batch correction papers is just like a grid of UMAPs, and they're like, look at how good ours did. And like this one is from what I would actually think is a good benchmarking paper, but like so I'm showing panel B and C. Panel A was actually like a table of the results, which is like, that's all you needed to show. And then they showed this one, which is just like, oh, look at how different the UMAP. Like, I don't know that this is any more integrated or retains biological properties better than this one. There's like almost nothing to be gained by having these side-by-side comparisons.
And basically what you've been saying for the UMAP stuff is they're pretty and they're qualitative, but they're not quantitative.
Yeah, so like
So
this
you can't
paper
look at
was a
something like this.
benchmarking paper trying to basically, it's like, I like this paper because it was not like, oh, we made this new method and this is why ours is the best. They're like, let's look at all the methods and figure out which one's the best. So I like that. but this is just like super misleading, I think, and it's kind of trying to tell people that this is how they should assess batch correction, is to make this grid and then point at one, which is not okay. I think that's just like super misleading. I think, and this is from last year, so like I think some of these examples are a little older. Like this one is from 2019, where like this was a main figure in their paper where they're like, okay, here's our data set, and this is a t-SNE plot, so it's a little bit different, but kind of the same as the UMAP. They're showing like it colored 3 different ways. Like each dot over here is colored by the biological sample of origin. The middle one is colored by the cluster or cell type assignment. And the right one is colored by like disease status, whether it was from a control or multiple sclerosis patient. And I think like, okay, like you might be like thinking like, oh, okay, like they're like in transcriptomic space, like they're totally different. Multiple sclerosis and control are like completely separated, but you don't know how many like points are in this plot, and you don't know what order things are plotted in, because they could actually go like right on top of each other. And then it could look like there's more differences, but there aren't really. And then like I don't even know like if there were a lot of differences, like there's t-SNE level differences in multiple sclerosis versus control. That's like not a real result that's worthy of being published in Nature. I think the goal they're trying to get to is like the biological differences between, you know, in their disease in different cell types, and like this doesn't help you get there. So like this is not the right data viz for this. I think the one that's like useful is like, I guess the middle one, just as the like bird's eye view of your data set, but you don't really need to go into like coloring it a million different ways. There's better things you can do, like stacked bar charts are honestly a lot better than that. Okay. Almost done, but I will go into like a little bit about trajectory analysis. So this is from a like very landmark paper in single cell field, RNA velocity in single cells. And this is like I don't know, a lot of figures from like 2018 or 19 in Nature are like a whole figure of just UMAPs, which is bonkers to me. But they're like, okay, we have this like, they had like, this is from like, I believe 1 sample. So there's no like batch effects or anything. It's just like brain development basically. So you have like these progenitors and then a couple of different cell types that they kind of like tips. And then, you know, the RNA velocity model is trying to build basically a trajectory based on the relative abundance of like spliced and unspliced reads. And then the typical like visualization with this is a vector field on top of a t-SNE or a UMAP. And then I think this like panel C is really compelling, where it's like, okay, like we see there's like, you know, the cells that are over here in the progenitor zone are like splitting off into these different branches. Which is like, I think that's really compelling. And like, maybe it makes sense in this case because it's like N of 1 and there's not all these other problems that are going to happen with dimensionality reduction, like, you know, things splitting by technical variation. But then again, like there's so many hyper parameters here. We don't know how long they messed with this until they got this like nice little thing with these like little arrows going, or like these little branches, right? Like it's not clear to me like how this would hold up if you like tried 100 different UMAPs with like different random seeds or different parameters. And the other problem with trajectory analysis is that you can literally apply it to anything. So like this is from the same MS paper that I was showing earlier, which is like a really good data set, by the way, but I think they had some problems that like the field has kind of gone past by now. But people might look at those papers and be like, oh, I should try this. Where this is like one cluster that they did like subclustering analysis on. And once again, like I think that their goal is to kind of like show you what's the difference between control and MS in this cell type. But the way they did it is they applied monocle 3 pseudotime, which kind of builds a trajectory like on top of your UMAP or t-SNE, but it's not the same algorithm as RNA velocity. The purple is basically the start, and then the yellow is the end. And in this one, it does look like, I guess you can't really see it, but like it's starts kind of in the middle, and then it kind of goes like around, but with 2 different branches, right? But then when they like do this weird plot over here, where like pseudotime is this axis, they don't really care about the branches, where like, you know, pseudotime 0.5 is actually happening in 2 different branches, so that it's misleading in that way as well. But I think that they're trying to say like, oh, okay, control is like enriched on this side and chronic MS lesion is on this side. So that means we found like that there's a transcriptomic difference. But this doesn't tell us anything about like what genes are changing or what transcription factors are involved. or like where is this in the brain? Like, you know what I mean? Like it's not really giving us anything.
Regardless of like the underlying things, like with the pseudotime issues, the viz on the right is also like not the best way to show this. Like use a line plot or a KD plot for something.
Yeah,
like
this is
this.
just
You're not
A
getting
jitter plot, basically,
quantitative.
right?
Yeah, exactly. This is pretty
Yeah,
weird.
I think like these are all very non-quantitative. So there's like this other thing people do, which is like they just make a UMAP and they add arrows to it.
Oh, there he is.
They so also like notice the journals, they're all nature or science or whatever. Like this is people like so the reviewers of these are often not bioinformaticians. They're just they're biologists and they're also misled. So like I think we need to be like the stewards of good data science and to not mislead people. I think that it's getting better and people are talking about it more these days. But like this one, like this is like an embryogenesis data set and they have like different embryonic time points. So like red is like the early time point, purple is the late. So they saw there's like this kind of like wave, right? And that's the way they like in Adobe, they drew the arrows, right? Which I don't think is even really trajectory analysis, but they're trying like this paper has like their main figures are pretty much just like walls of UMaps. So it's like this was yeah, it is what it is. But I think I'm also like going to beg the question of like, is a third UMAP dimension adding anything if like all of our media like papers and presentations are 2D? There are actually like I don't know if anyone's tried it, but there's like VR apps for going inside your single cell data set, which is like crazy. And I guess in that case you would want it 3D, but I don't know why you would want 3D and then you're like moving it around to find like the best angle. Like it just seems really like unnecessary to me. Okay, we've reached the end. So my final like summary of this is to always visualize your data and try visualizing in different ways. Master one of the Python or R plotting libraries and have a somewhat of a working knowledge of at least one other one. Learn the basics of a graphic design software like Illustrator or Inkscape. I didn't mention it earlier, but there's a million YouTube tutorials for those things as well. So if you're like, how do I align something in Illustrator? YouTube will have the answer. Sometimes they're like more oriented for like marketing people because I think like graphic designers, that's like their daily driver software. But I think it's still very useful. Yeah, always be thoughtful when you're picking color schemes and you're figuring out spacing between panels and use of white space. Remember the classic pitfalls in the friends don't let friends GitHub and don't abuse dimensionality reductions as we were talking about. I'm also going to suggest that if you find a figure that you like, you should take note of it and try to think about what they did right that you liked and how it conveyed the idea correctly. And then on the other hand, if you find a figure you don't like the banana one that I found, I have like a work going like Google document that has like bad examples. I didn't show all of them, but I was like the banana is going straight in there. I think that I do like the bananas. Like it might be so bad that it's good in some way. I don't know.
Yeah.
And then I think also if you have a really bad figure and your paper's still in Nature, that means your science is probably really good. You don't have to hide behind a fancy figure.
They don't-
But if you can, then you're more likely to get the distinction.
I think people like things that look good, like subconsciously. So like make things look as good as possible, and that will you'll probably have some
buoyancy.
some benefit I think will occur. But yeah, it could like sink a lot of your time. So also don't dwell on it too much, especially like for example, if you're making supplementary figures, like some of mine are just like I break every rule because it's just like it just needs to go in and I don't need to spend a million years on it. Okay, that's it. So thank you all for listening.